Good day everyone, I am Angela, a 3 BVMD student. Our topic for today is about opioid analgesics. So first, here are the table of contents for today's topic. The first is the physiology of opioid. The second one is the pharmacodynamic of opioid. The third one is the opioid pharmacokinetic. And the last one is the clinical pharmacology of opioids. First is we're gonna talk about opioids and its physiology. So poppy seeds or poppy are a plant, of course, that grows as you know. So the derivative of poppy seeds are opiates. And opiates are chemical drugs derived from poppy seeds. Well, such as heroin, essentially and chemically. Okay, once that we've created to synthesize the effect of opiates, those are called opioids, which bind to opioid receptors. Example of opioids are natural opioids such as morphine and codeine. These are also known as opiates. Semi-synthetic opioids such as heroin, oxycodone, and hydrocodone, which are chemically altered opiates. Fully synthetic opioids such as fentanyl and methadone. So, the use of this drug is to reduce the pain. We often use them in combination with anesthesia to reduce the amount of anesthetic needed. And originally, oddly, who knew but opioids were used to treat diarrhea during those days of cholera and dysentery. Unfortunately, opioids are extremely addictive. They are controlled drugs. We have to be very careful about our stockpile or stock counting of opioids because if any go missing, we technically have to alert the police because taking in high doses, they can be deadly. Do you know that we have three classes of opioid receptors? The receptors that we're talking about opioids are the mu receptor, kappa receptor, and the delta receptors. We tend not to talk about delta much at all. We really need to focus on mu receptors and kappa receptors when it comes to opioids. So the first one is mu receptors, create the most analgesic receptors or opioids that are consistent with mu receptors or our mu receptors base. They are really known for their amazing level of analgesics. Likewise with kappa receptors, they also known play into the analgesic drugs like kappa susceptible receptors as well. They are also known for their sedation. For delta receptor, it plays a role in analgesia and reduction in gastric motility. So, opioids have presynaptic and postsynaptic effect. Presynaptic, they prevent the release of the neurotransmission, and for postsynaptic, they kind of create that inhibitory effect, so to prevent more of those pain receptors from depolarizing. Little bit of review. So we're going back to physiology. I'll just show you here. We've got this in the picture of the inside of the cell. This is the outside of the cell, and this is the plasma membrane. Okay, with the hydrophilic and hydrophobic ends, so the receptors technically kind of sit within the plasma membrane because then they connect to the other side and then they allow the drug in. So here's another good example of chemical signaling. So this is the binding sites that fit a specific shape. That's the protein that sits with the plasma layer. It accepts only the specific shape, which is the opioid. So this is the opioid receptor, and then it allows it to have an intrinsic effect on the cell. So it allows the effects of the opioid to then react to the inside of the cell. Opioid receptor at work. We have three opioid effect. We have agonist effect, antagonist effect, and a partial agonist and antagonist effect. Let's see the opioid receptors at work. So how they work. So how an opioid receptor works. So here we've got hydromorphone. This hydromorphone molecule, it fits perfectly with the opioid receptors, and it has a high intrinsic value. So that means that it has a high intrinsic effect on the cell. So this is a pure agonist. It fits perfectly and it has an intrinsic effect on the cells. Then we have this opioid receptor and we have amoxicillin molecules floating around. As you can see, they don't even fit together. So that's not going to work and there's nothing going to happen. No effect. The drug doesn't even match the receptor shape. Then we have this guy, butorphanol, who is a partial agonist-antagonist. It fits to the opioid receptor really well, but it has a less intrinsic effect than hydromorphone. So, hydromorphone, you're going to get tons of long-lasting pain relief. You're going to get respiratory depression, and the animal might even be a little bit of sedate, but not much. So, let's move on to the analysis of those three, the agonist, antagonist, and the partial agonist-antagonist. So that we've got totally 100 mu agonists, so working on the mu receptors. We have amoxicillin, which isn't even going to combine with the receptors. So let's just show you the difference in shape. And then we have this guy, which is a partial agonist and antagonist, which fits the receptors, but it has a lesser effect. So it's actually, in a way, reversing the effect of the hydromorphone. We will give the three opioid receptor an example. For the mu agonist, we have selected here, the first are the endogenous peptides that have the highest affinity for mu receptor. These are the endorphins, mainly beta endorphin. It has the highest affinity for mu receptor and lower for delta and kappa receptors. The second is an exogenous drug. The drug is morphine, and it has a greater affinity for mu receptor than kappa and delta receptors. Naloxone is a drug in clinical use that acts as antagonist of mu receptors. Please note that the naloxone icon that makes almost a perfect match with the mu receptor. This illustrates that high affinity for the receptor. Delta receptors in the slide will see the molecules that act as agonists of delta receptors. First, the endogenous peptides with high affinity. These are the enkephalins. They also bind with the mu and kappa receptors, but they have high affinity for delta receptors. The exogenous drug that acts as agonist of delta receptor is the DPDE. This molecule is used for research purposes. The drug that acts as antagonist of delta receptor is the same done for the form receptor naloxone. It also blocks delta receptors, but if you pay attention to the icon, as you see, you will see that there's another perfect match at the binding site. The intention is to show that naloxone has a lower affinity for delta receptors than for mu receptor. The last classic receptor we are going to discuss now is the kappa receptor. As with mu and delta, we will study its main agonist. The endogenous opioid peptides with the highest affinity for kappa receptor is dynorphin. Dynorphin has also some affinity for mu and delta receptors. Ketazozine is the name of the exogenous drug with highest affinity for kappa receptor. It is a drug used experimentally for research purposes. Finally, let's find out which drug as antagonist for kappa receptor. We can see again that naloxone binds to kappa receptors, but if you take a closer look, you'll see that the naloxone icon doesn't match perfectly the kappa receptor. So what the diagram wants to show is that naloxone has a lower affinity for kappa receptor than mu receptors. So it's Abby's turn for the next subtopic in opioids, which is the opioid pharmacodynamics.